Hello, my name's Alan Pang. I hold a MSc Mathematical Science distinction, and through these couple months research onto the COVID 2019 and its implications, and which has caused detrimental and economical impact to the pandemic, on which has arisen due to R0, on which R0 is the basic reproduction ratio, on which this was associated with the Wuhan in the Wuhan market in China, in the Oriental market, and that it liaised. But there's enough evidential data to suggest that the genomic structure, which was analysed from the Wuhan, found that it came from pangolins, on which there is association of the human genomics associated with the human genomics code, associated with the coding in the market in the Wuhan market in China. So there's the association that this coronavirus is a different strain of the variation similar to the genomics from the 30 sequence genomics by researchers in China and Hong Kong. So in on which this was publicized in April, round about April, this was publicized on which scientists has measured the genomic sequence, on which they say that SARS is similar to SARS of its family, like Middle Eastern Respiratory Syndrome, on which SARS has an efficacy, on which I have analysed that is similar to it's similar to corona and other rotaviruses, but different entry, like entry pathway through something called hemagglutinin. On hemagglutinin enters the surface of the cell through endocytitis. On the hemagglutinin glycoprotein attached to the cell, but on which the SARS-CoV 2019 has engaged effectively with the extracellular cell membrane through endocytitis of the ACE2 interaction of the S spike protein attached to the nuclear capsid and the RNA molecular structure. So when the hemagglutinin has attached via the hemagglutinin attaches via the glycoprotein, on which it gains entry on the extracellular of the surface membrane through something called N-salicylic acid, but in association with the NU9 salicylic acid enzyme. The other one is the association between ACE2 enzyme, and ACE is an angiotensin converting enzyme associated with the zinc carboxypeptidase, which entered through that cellular entry, and then increasing the distribution of the ACE is through elevated sodium levels of excretion from the kidneys and the potassium levels. So it enters through that respectively. So what I've understood from my research is that basically the behavior is different, but antiviral properties do not work against hemagglutinin of the HA and HN1 or H5 or N1 to hemagglutinin, and numerindase is the cleavage away from the salicylic acid towards infecting other host cells after transition period and the translational period. So what I've analysed is that basically it behaves similar to like antiviral properties like Relenza and Tamiflu has no association. Did not form any association between the sleeve due to the molecular structure. The molecular structure is not similar because antiviral acts as like a conjugate of a conjugate pair of a different variation of the salicylic acid, on which this is a the importance of the recognition of antiviral interventions of Tamiflu and Relenza is not efficacious effective against the COVID 2019 due to different structure formations and different viral entries, basically because the S spike protein attaches to the ACE, which associates with the TMPRSR22 protein of the proteacelia through endocytitis. So when the S spike protein of the glycoprotein attaches to the extracellular cell due to something called a conformational change of pH, allows it to enter the cell respectively. So basically the association between identification between um, the the H1N1 and SARS is different viral for the M protein interactions and channel gates on. So due to a proliferation and conformational change allows the glycoprotein to enter the cell to attack inside the endothelium of the alveolus of the cell, to which engage in the transitional period of entering something called an exosome and an endosome. An endosome or lysosome pH, where it encloses itself inside basically of a protein capsule, on which due to conformational differences in pH of acidification of a high acidification level, it allows the release of the viral entries, the viral genomes and the antigens into the transcription and translation phase needed for photocopying. So scientists have worked efficaciously to prevent basically ACE2 by on which they have scientists and researchers had utilized things like angiotensin converting enzyme inhibitors and prohibitors from preventing the cellular entry and association with, on which is dangerous because basically the things associated with the different things like Lanzapro allows different things like rheumatitis interventions and rheumatoids, or things like acute myeloleukemia EGCG and Cachichin, which associated with things like BCL kinase of adenosine triphosphorylation of the energy associated with the interactions through coffee up regulations of the glycoproteins and the amino acids sequence on utilization of mesylate and other things like RBS. But scientists associate this with this affects the utilizations of things like blood regulations and blood sugar, blood clots, and association with reduced increase in hypertension and increase um, in in cholesterol levels and BP, blood pressure, because blood pressure is to do with vasodilation, vasoacromic prostaglandin, which are associated with ACE and bromophasin and in the endocrine system. So when the ACE, the angiotensin, no, angiotensinogen converts ACE121 to ACE127, associated with the kidney functions, the filtrations of associated with filtrations, an increase in pH of vasodilation and vasoconstriction through prostaglandin and PGCA release. So these affects things, these relatively affects the ACE, it's the interaction with the ACE on interactions with things like inhibitors like olanzapine and lonastatin and other kinds of interventions were associated with antiviral remedies against preventing. So preventing the ACE inhibitor had utilize things like immuno antibodies, on which I believe that antiviral remedies have greater efficacy against COVID 2019 from my understanding and my research. So I believe that ACE has this association with high cholesterol, and from observation from the media and the patient on which was submitted into hospitalization, is to do with high cholesterol and increase in BMI, which is body mass index. So increasing in BMI increases the chance, because once you and there is association with increase in anxiety suppresses the immunity system in our bodies, that associates with increased risk and exposure to COVID 2019. On which so if you increase the because there needs to be enough in receptors for engagement through the interactions of the COVID viral antigen attached to the cell for cellular entry. So increasing the distribution of the ACE increases the chance of propagation into the cell for translation, transcription, and photocopying, on which it hijacks the viral machine mechanism of the transcription phase and translation of the sequencing of the amino acids from the copying of the DNA RNA of the GTAU, which is guanine, thymine, adenine, and uracil for RNA of the nucleotide analogs. So and scientists has utilized things like HIV medications of adenova, aciclova, and other HIV, which utilizes nucleotides, the nucleotide analog, which attacks the RNRDP progression or transcription to prevent viral transcription and viral copying. So these are the things, the latest interventions against antiviral replications, on which I want to suggest that even though with these antiviral engagements of hydroxychloroquine and things like remdesivir, there is susceptibility that it interacts with zinc. On the zinc needs to intracellular, acts as the hydroxychloroquine acts as an ionophore through the RNRDP of the transcriptase of the 3' to 5' of the translation mechanism. So this is very important in understanding these procedures to allow they allowed efficacious fundamentals between the photocopying and reducing to the increasing. So hydroxychloroquine acts the hydroxychloroquine with the zinc and the acts as the ionophore, which transcribes the cell, the zinc intracellularly, extracellularly from the endothelium of the cell internally inside the cell to allow the transcription phase to occur. So this blocks RNRDP, which is RNA dependent polymerase of the enzyme for the photocopying of the transcription phase through the promoters and the primers. So through things like messenger RNA, which is riboxynucleic acid of the nucleotides. On nucleotides are sets of DNA of the human genome, through which is sequenced from pyrimidines to purines, from adenosine and aciclova, adenosine, guanine, and cytosine, and thymine, and uracil for RNA, which is made of a benzene ring, benzene ring with the oxygen molecule attached and amides groups attached to it. So which something called so for viral entry or viral copying for the functioning of the viral activity, as it's very important as the human mechanism to something associated with ATP and adenosine triphosphorylation, which is needed for energy. And phosphorylation is the addition of a phosphate group. So this is very important. So blocking this prevents the viral copying, and it blocks viral replication. So this reduces, as there has been studies from researchers that this has reduced the number of the viral load from. So the additions of these antiviral interventions is important of the efficacy of like they're saying that the release of COVID vaccines. COVID vaccines uses immune antibodies secreted from small fragments of the viral genome, which is secreted inside the body to allow the immune response to attack the natural killers and the cell mediate to initially recognize this, on which there has been association on which the COVID 2019 affects coagulation due to over addition of something called ARDS, which is acute respiratory distress syndrome of a accumulative immune cytokine storm. So these are all the interventions of COVID, and there's been association with intense acute respiratory distress syndrome and organ failure due to pneumonia caused by bacterial infection. On fixing things love, azithromycin is like a macrolide which prevents transcribal transcription translation during the ribosome of the translation process of the DNA. So these are very important efficaciously, but it's important to not to utilize the interventions of too much reliance on too much of the vaccine to prevent COVID 2019, as there are variants of cumulative load of variants of the genomic structure which needs to be identified. So what I suggest more efficaciously, due to the scientists working on the antiviral remedies to attack COVID 2019, the suggestion is a proficient algorithm that allows us to understand how to understand and detect the COVID strain through algorithmic procedure of detecting the genomic sequence. So the genomic sequence is made up of understanding what ATTA and in the sequence, which makes up the amino acid space basically, and needed for transcription and the viral copying through slicing and different various of biomedical bio mechanism and procedures that occur in our bodies. So after this, so the most important part is what scientists are working on is to reduce the viral load of increasing lysosomal pH of the hydroxychloroquine. And hydroxychloroquine acts as the iron 4, which transfers the zinc to prevent RNADP polymerase of the transcription phase. So what I would utilize is because they are there's increased worrying concept along with the government alignment with due to understanding of R0 as well of increasing susceptibility of risk to the vulnerables is to identify the genome of the COVID strains and to segregate them respectively through algorithmic procedure on which to identify the relative genome in the sequence to identify the number of counterfeit AA a number of adenines and thymines, uracils, and other cytosines and guanines. So to understand these, to quickly identify in comparison the genome of the human structure allows us to counteract this problem respectively. So what I believe that in the future that hopefully there'll be more in research into antiviral properties of things like the utilization of remdesivir or hydroxychloroquine or chloroquine or utilizing catecholamine and EGCF on which chocolate or things like cocoa and other antiviral properties from plants on which individuals that utilize that dark chocolate made from cocoa has antiviral interventions on which has high concentration of zinc, on which zinc plays an important role inside of body of preventing COVID by blocking RNADP in the body. Thank you.